අපි මේ ලෙසන් එක ඉවර කරේ මෙතනින්. එතකොට මෙතනදි තියෙනවා අන්තිමටම එකක් ඉගෙන ගන්න තියෙනවා. ඒක තමයි non-inherited characteristics. What are non-inherited characteristics? The characteristics which appear due to the changes of the environment, nutrition, exercises, and activities are not inherited. Think like someone went to the um, gym and they build their body, they build their muscles. So this characteristic will not go to their children because it is something which they have gained by exercises and by sometimes by nutrition and the environment. So they are not inherited. Okay, example, growth of muscles by exercises, cones in toys and scars on skin. Okay, so here we are going to discuss about this Gregor Mendel and Mendel's experiments. I have told you, I think we've already discussed this thing, but I am going to tell you it again. The first person to study about the transmission of inherited characteristics is an Australian priest and a science graduate, Gregor Mendel. He is honored as the father of genetics. So he is the father of genetics. Highlight it. This is the Gregor Mendel. Mendel's experiments about inheritance. He used the garden pea. What is the plant he used? It's garden pea or Pisum sativum. The scientific name is Pisum sativum and the other name is garden pea plant for his experiment in 1865. Okay, he has used that particular plant for his experiment because of its special features. You should know the special features of this plant. Okay, the reasons to select the garden pea plant for his experiment as follows: easily grown, so highlight these things. Easily grown, obtain the yield within a short period of time. Yield within a short period of time and obtain pure breeding plants ගන්න පුළුවන් වෙන එක can obtain pure breeding plants. The selected characteristics are not changed for many generations. And the next one is naturally self-pollinating. When necessary, cross-pollination is also possible. Naturally, it has adapted to self-pollination, and when it comes to artificial pollination, that means they could do with those plants we could do cross-pollination also. Okay, and ability to obtain offsprings by breeding and can continue the generation. Ability to obtain offsprings here, offsprings by breeding and continue the generation. And there are 7 contrasting characters and he tested one character at each time. Tested one character at a time. The procedure he followed for the tall and short character was as follows. So there are 7 characters. We have them in this page number 175. You can see color of the flower, color of the seed, shape of the seed, shape of the pod, color of the pod, position of flower, height of the plant. So here we are going to discuss about this height of the plant. From this example, we are going to learn this method, Mendel's method. And other than this height of the plant, we have color of the flower and position of the flower. Color and position of the flower, color and shape of the seed, color and shape of the pod. So these are the 7 characteristics we are going to learn, and there are dominant characteristic and also recessive characteristic. What are the dominant ones? The height. When it comes to height, the tall ones are the dominant. The position of the flower is axial. The color of the pod is green. So these are the purple, yellow, round, inflated, green, axial, tall. So these are the dominant characteristics of these characters. Dominant means that is the one which shows when you have 2 dominant and recessive genes, it also shows the characteristic of the dominant instead of the recessive. Okay. එතකොට අපි example එකක් විදිහට ගත්තේ we are getting that height. So that height, if we use capital T for the tall plants and simple T for the short plants, we could end up with this table. I'll show you it, and I think we discussed it already, but I'm going to tell them again. Okay. So the procedure he followed for the tall and short characters was as follows. So you should know the Mendel's why Mendel selected this garden pea or pisum sativa plant, and what are the special features which lead him to select this plant for the experiment. Okay, so we have 1, 2, 3, 4, 5, 6 reasons. So you have to remember them all. Cultivation of pure. So now we are going to discuss about the procedure. Cultivation of pure breeding tall and short plants. Pure breeding tall and short plants. This parental generation was known as P. So this is parental generation and we name it as P. Pollens of the tall plants were deposited on the stigma of short plants and vice versa. Okay. So the pollens of the tall plants also deposited on the stigma of the short plants, and the pollen of the short plants were deposited in the stigma of the tall plants. You know what is pollen, what is stigma already, because we have done that lesson earlier. After that, obtain new seeds by cross-pollination and plant them to obtain the new generation. All the plants were tall and they are referred to as F1 generation. So here this is P generation. So pure breed plants. So this is capital T. Capital T means the both genes are dominant, and here both genes are recessive. So this is pure tall plant and this is pure short plant. So the gametes comes from here is capital T. Anyway, this, if you take the left side T, it is capital T. Then the right side T, it is also capital T. When it comes to recessive genetics, we only obtain simple T. Okay, there are no any dominant gene. So the gametes coming from capital T, T is capital T. The gamete coming from simple T, T is simple T. So from the pollination, from their after the pollination, we get this F1 generation as capital T, simple T. It should be like that, no? Because we are getting here capital T and from here simple T. So the F1 generation, all the F1 generation should be like this, capital T, simple T. And then we come to the, so here see the P generation. This plant is height and this plant is this, so capital TT plant is tall and simple TT plant is short. So after we take two gametes and after the breeding thing happens, what happens? We are getting F1 generation as capital T, simple T. Their gene represent as capital T, simple T, but this capital T, that tall plants are the dominant ones. So the dominant characteristics of capital T is indicated from the plant. So the plant is tall, but it has that recessive gene which have the characteristics of short plants, but it does not show. That capital T does not allow to show the characteristics of the simple T. Okay, because capital T is the dominant one. I think you got this one. After that, we are getting F1 generation like this. All are tall plants from their appearance, but they have genes which bear both tall characteristic, the capital T, and the short characteristic as simple T. Okay, so when we take the gametes of these two plants, when we are going to do the breeding or pollination of that plant, we have to consider about their gametes. When we consider about the gametes of F1 generation, you can see that it is capital T and simple T. It could be capital T or it could be simple T. So yeah, so see, sometimes it could be capital T and sometimes it could be simple T. We don't know what will it ended up, in which type of gamete. So we have to write both, and here also we have to write both. So this is a kind of parent plant, because F1 we are going to make a F2 generation from F1. So P generation, it gives F1 generation, and from F1 generation we are going to make the F2 generation. When it comes to F1 generation, see, we got two gametes, and we are going to do the cross pollination among them. So here you could go with this capital T in the second plant, or this simple T in the second plant. If you go with capital T of the second plant, you are ended up as TT. So what is the character, the appearance character of this plant? Is it tall or is it short? It has capital T and capital T. What is the characteristic? Yeah, it should be tall plant, because it is a pure tall plant, no? Which was there in the beginning. So it is like a pure tall plant. And if this T go with this simple T in the second plant, so it ended up as capital T simple T. So the appearance, the character it gives is the height, but what is the difference? But it has both dominant and recessive gene inside it. It only shows the characteristic of dominant gene. Got it? Because it is TT. And what about the simple T? So after the simple T goes with this capital T in the second plant. The first plant simple T and the second plant capital T, but it ended up as this capital T simple T. So the appearance is tall, but it has both genes. And what about this one? If this simple T goes with the simple T in the second plant, you are ended up as simple T. So the appearance is short plant, because all the simple T's are recessive. It doesn't have any dominant one, or it doesn't have two pure breeding tall genes. Okay. එකේ තියෙන්නේ pure තමයි, pure short genes. short කරන gene එක recessive එක තමයි තියෙන්නේ. Okay. So here gametes, we are going to draw this kind of we call it as Punnett diagram. So this diagram here, we indicate the female, and here we indicate the male. So the female plant is, so one plant here, one plant here. So see, this female plant bear T gamete and capital T gamete and simple T gamete, and this side male plant also bear capital T and simple T. So this is the F1 generation, and we are going to write down the F2 generation from these two. So capital T, capital T, and the characteristic is tall. Here capital T, simple T. So the characteristic is tall, but it has recessive gene too. Here capital T, simple T, appearance is tall, but it also has two genes. One is dominant and one is recessive. And here it is simple T. That means it do not contain any gamete which shows any gene which shows the tall characteristics. That's why it has to become a short plant, because it do not contain any capital T dominant gene. Okay, it has only simple T, simple double T gene, which is considered as a pure short plant. Okay, so that's all about this one, and we are going to discuss it here again. See, pollen of tall plants were deposited on stigma of short plants and vice versa. We discussed. Obtain new seeds by cross pollination and plant them to obtain the new generation. All plants were tall, and they are referred to as F1 generation. So even though we mixed that tall plant and short plant, all plants were tall. All plants were tall. Remember that part. All plants were tall. Allow self-pollination to take place within F1. Within F1, what is their purpose? They allow the F1 generation to self-pollinate. And we are going to check the ratio after the self-pollination. The resultant seeds were planted to obtain the F2 generation. The tall to short plant ratio was 3 to 1. 3 to 1 is from their appearance. See, 3 to 1. The three plants are tall because it contains dominant gene, and one plant is short because it does not contain the dominant tall plant gene. Okay, so from the appearance it's 3 to 1. Yeah, from the appearance it's 3 to 1. But if we consider about their genetic material, so if we consider about genetic material, only 1 to 1, the ratio should become 1 because these both are dominant 1. So, and this is same, so 1. Here we get 2, then ratio again, simplicity, it's 1 to 1 from their genes, but from their appearance it's 3 to 1 because these all three are height and tall, and this one is short. So from the appearance it's 3 to 1. Okay. Then it was a question to Mendel that what has happened to the short character if one it was a question to Mendel that what has happened to short character in F1 generation. According to Mendel's opinion, the tall character was dominant and short was recessive. Mendel has used large number of samples and repeatedly done the experiment to confirm his conclusions. Analyze how Mendel has carried out his experiment according to scientific method. The feature which was recessive in F1 generation reappears in F2 generation. That's right, no? So in F1 generation we didn't see a short plant, but when its recessive gene goes with the recessive gene of another plant, so then they become tall, not tall, short. Yeah, so even though it doesn't show its real characteristic in the F1 generation, it has to show it in F2 generation sometimes. Okay. It was an important observation as Mendel used a single pair of contrasting characters at a time. It was known as monohybrid cross. So what was Mendel used? A single pair of contrasting characters at a time. So these are the contrasting characters, the 7 characters. So he used one character at a time. Color of the flower, so he made this purple and white. Then he ended up with F1 generation purple. So he decided that because of that F1 generation is purple, this purple color is a dominant color. And here color of the seed, yellow and green, the pure yellow and pure green is breeded by cross pollination, then ended up with yellow color seed. So that means the yellow color of the seed, the color yellow is the dominant characteristic, dominant feature. So other one, the green is recessive. And after you have self-pollinated this F1 generation, you are getting this much of yellow and this much of green. So the ratio is nearly 3 to 1. It's self-pollination, that's we are getting nearly 1. So this is the one. From here you can identify what is dominant and what is recessive gene by doing this experiment. Okay. So they were called as monohybrid cross. The inherited pattern of a monohybrid cross. The results obtained from the Mendel monohybrid cross using 7 different contrasting characters are given in the below table. We discuss it. Okay, we are coming here. Okay, I have already told you these things. It is clear that the above characters are inherited in the same manner. One feature is completely hidden in F1 generation and it happens in F2 generation. This is the recessive feature. In both generations, all the plants were with the features of P generation. No P plants were with intermediate characters. Mendel assumed that it was because two different factors determine a single characteristic. In genetics, symbols are used to denote those factors. The standard is that the dominant factor is denoted by a capital letter and the recessive by a simple letter. So here, accordingly, for tall feature, it's capital T because it's a dominant characteristic, and for short feature, it is simple T because it is a residue character. Sometimes it could be changed, okay, sometimes it could be the short feature could be the dominant characteristic, then you have to use the capital T. So here we are practicing, we use the capital 1 for the dominant characteristic, okay. For each inherited feature, two factors are involved: pure breeding tall plants, capital TT; pure breeding short plants, simple TT. The tall plants with recessive short feature is capital T, simple T. Okay, when two factors are similar, they are known as homozygous. Homozygous. That means if they are capital T or simple T, they are considered as homozygous. When the factors are not similar, they are known as heterozygous. That means capital T, simple T. So homozygous and heterozygous. Okay. The monohybrid cross of pea plant, considering tall and short characters, can be expressed using symbols as below. Punnett square. Here we have a Punnett diagram, Punnett square. So in exams, sometimes they are asking you to draw the Punnett square, which was introduced by scientists called Punnett, can be used to show the occurrence of F2 generation. It is the way how we are going to draw the F2 generation. See, F2 generation is created by using F1 generation 2 genes. Okay. After that, explanation of patterns of inheritance using probability. So I'm going to discuss it later. For now, I'm going to stop here and we are going to discuss it later in the next part. Okay. Thank you.